Next one is Alana Ferguson from Suncoast. And so when I was reviewing the application, Alana's resume obviously popped up because she is the number one at Suncoast High School, that if you know is a very tough high school. So when I set up the interview, I had this idea in my mind about Alana. Oh, she must be a bookworm. She must be always there studying. And actually, I have to say, I could not be more wrong than that, because Alana, she's not just an amazing, talented student, she has also a ton of interest. She's a runner, and I love that about you. She loves to read, that you do drawing, and also I heard that she was the one doing the best stitches during our stitches workshop on bananas. So you also have that skill, so you're really an amazing all-around student, and we are very glad that you came here for this internship. So she is the next two students, so Alana and then Sam, had project connected to the Fitzpatrick Lab. So please.
Okay, thank you so much, Ilaria. My name is Alana Ferguson, and I am a rising senior at Suncoast High School in the Math, Science, and Engineering and International Baccalaureate programs. And for the duration of my internship, I was working out of the light microscopy core, testing long-stokes shift dyes for nanoscopic visualization of synapses. Now, in order to break down that title, we first need to look at some of the background information associated with the samples that I was looking at. So here at the Institute, we have the Fitzpatrick Lab. And the Fitzpatrick Lab looks and investigates the cerebral cortex with specific information being gathered from the visual cortex. So the visual cortex processes sensory information that we're looking at through our eyes in order to make connections between different neurons so that they can communicate with one another. Our cells respond differently to different visual stimuli. And here at the Institute, we're studying the different ways that cells differentiate between these stimuli in order to form connections with one another. Now, in order to look at these connections, we have to look at a fundamental communication unit in the nervous system or in the brain called synapses. And so I'll be looking at 4 different components of a synapse in order to determine those areas of connection between different neurons. And so if you look at this image here, you can see 4 distinctly different parts of a synapse, but you can't necessarily distinguish between them. Everything's black and we don't know what is labeling what. So here at the Institute, and a common practice that a lot of people do for different research projects is stain these samples and target specific structures with fluorescent proteins or fluorophores. So if you look here, we have different fluorophores targeting our other structures. So we have this blue one here and this orange one here. And what we will do is we will shine a light on it, an excitation beam, and that will cause these different structures to fluoresce or release light so that we can go ahead and differentiate between them. Now, when I'm looking at the synapse, I'll be looking at these 4 structures. So I'll be looking to differentiate between the axon, which is the area of a synapse or a neuron that is sending information to another one. And then I'll see this presynaptic protein that will be attached to the axon, which will be one of the markers. I will also see this postsynaptic protein here attached to the dendrite, which is an area of another neuron that is going to be receiving the information at that synapse. Typically, we have specific dyes that we would be using in order to visualize these structures, but for the purpose of this experiment, we're actually going to be using a new type of dye called long-stokes shift dyes. Now, in order to understand what long-stokes shift dyes are, you first need to look at an emission spectra. So typically, you have different wavelengths and colors of light, and if you look here, you can see that on the high energy side, you have your colors like purple and your colors like blue. And then if you look at the lower energy part of the spectrum, you can see your colors like red and orange. And so imagine we have a sample that we're trying to look at in green. In order to visualize the structure in green, we need to make sure that it's emitting green light when we shine that excitation beam that I was talking about on the sample. Because some of the energy that we're shining on it is either going to get lost to heat or it's going to get absorbed by the sample, we need to shine a higher excitation light upon that sample so that it fluoresces in a color that has slightly lower energy. If we look here, we're shining this blue light, which is slightly to the left or has higher energy than the green, so that when some of that energy is absorbed or is transformed, we're fluorescing in green. The difference between this excitation here and this emission here, which is the color that we're fluorescing, is a Stokes shift. Now, with these new novel dyes, we'll actually have a longer Stokes shift, or a longer difference between the excitation and the emission of these dyes. So if you look here, we're still exciting it with something on the higher end of the spectra, or blue, but we're going to be emitting all the way in the red channels, which is going to be more efficient for our super resolution microscopy, which I'll get into a little bit later. So if we look here, this is the emission and absorption spectra of these dyes. And so this would be our control slide where we're using our typical dye configuration. If you look here, there's that one dye that I was talking about that's our typical dye. We're exciting it in a cyan blue channel, and everything else is going to get excited by the red. Now, the problem here lies within the emission spectra. If you see, we have these 3 dyes clustered together in red, and that will be very efficient for our super resolution microscopy. However, we have this one dye over here that's kind of differentiating itself from the rest, and that separation is not going to be efficient for our super resolution microscopy. It will make it a lot more difficult to differentiate between these different structures. So using the long-stokes shift dyes, we're actually able to still excite that one green structure with that cyan or blue range dye or excitation beam, and everything else is still being excited within the red end of the spectra. However, if you look at the emissions, you can see that everything is kind of clustered together, which is more efficient for a super resolution microscopy and will allow us to resolve those structures using those microscopes. Knowing this, I actually didn't go in using a super resolution microscope to begin. I went in using a confocal microscope. And so a confocal microscope allows us to take this wide image of the structures we're looking at so that we can differentiate between different areas and locate our regions of interest. If you look here, we have some of the dendrites being stained in green and our axons being stained in red. And then we have our presynaptic marker marking everywhere in the cell, but we have concentrations of it in specific areas. Locating this with the slice, I was able to determine that yes, there are synapses and there are points of contact between different neurons. And I was actually able to go in and start imaging within these specific regions to determine if we have neural connections here. And so taking a closer look on this confocal microscope, I was able to see more of the individual dendrites and axons, and you can see in even more detail those pre- and postsynaptic markers, marking that we do have connections between neurons here, and there is some communication going on in response to the visual stimuli that was looked at earlier in vivo or live. So these samples are actually fixed, so we're looking at it after the experiments were taking place. And so using the confocal microscope, I went in even closer, and I was able to visualize some potential areas of contact between axons and dendrites. So if you look here where the red and the green are touching, there is a potential that this is a synapse, especially since we have our pre- and postsynaptic markers surrounding it, but we can't necessarily differentiate between if they're crossing over or if they're actually touching, like Justin touched upon within his presentation. And so using this, before I moved on to our super resolution microscopy, I wanted to test those long-stokes shift dyes I was talking about in order to determine if they were attaching to the structures or binding to those specific structures I spoke about and working in the way that we were expecting them to. And so I used a method called multi-channel imaging or lambda imaging. And so what that is imagine you have a blue light that's shining on some sort of sample. We don't know exactly what the sample is composed of, so we don't know exactly what colors make it up. But when we shine this blue light on it, we are going to fluoresce in different lower energy color channels, and then those color channels are going to differentiate between different structures or regions within that sample. Now, applying this to my project, I first looked at the green structures that were being boosted by those long-stokes shift dyes I talked about. So if you look here, you can see these structures within the green that I was trying to replicate within the red channels, because the purpose of our long-stokes shift dye was to shift what we were seeing and emit it within the red end of the spectrum to be more efficient for super resolution microscopy. In actuality, this image was boosted up with the brightness tenfold or by a large amount so that you could visualize what is here in the red channels. We're actually not able to replicate, and we didn't expect this to happen, but we're not able to replicate these green structures here in the red, which is showing us that there's a potential our dye was not attaching in the way we were expecting it to. Because this green laser also has a lot higher energy than some of the dyes and some of the other structures we saw here, there's a potential that it's turning on other structures and lighting up so that we're looking at things that aren't necessarily what we want to be looking at. The same is true for our red channels or our red boosted structures with the long-Stokes shift dyes. So if you look here, we're actually trying to replicate these structures within the far red channels. In this image also, the brightness was boosted up for visualization purposes, but here, these are actually very dim in comparison to the structures we're seeing within the red range, also reminding me that there's a potential our dyes are not attaching to the structures in the way that we were expecting them to. This prompted me to take a look on our super resolution microscope, or the STED, which allowed me to view these structures with a lot of detail to determine if there was synaptic contact and if these structures and if these dyes were being targeted specifically. And so first I had to take a look and learn the difference between confocal and super resolution microscopy. If you look here, this is the cytoskeleton, or that supporting skeleton in the inside of a cell. And so here you have tubulin and actin, which are different filaments and proteins. And if in the confocal microscope, you can see that, yes, we can distinguish between some of these structures, but we're not actually able to go in and differentiate completely whether or not there's 2 or 3 filaments there or what's crossing over with what. So we can see that there's something there, but we can't see the specifics of certain things that are going on. Using the super resolution microscope that we have here in the Institute, or the STED, stimulated emission depletion, I was able to go in and actually differentiate between these individual filaments. And if you look at this region here, you can see it with much more detail and differentiate between those structures. I spent the introductory period of my internship learning how to use the STED, or super resolution microscope, because there's only about 10 of them in the United States and less than 50 people in the United States who know how to use this microscope. And so my mentors helped me learn how to use it and learn how to correctly operate its parameters so that I could go ahead and image the structure and image the sample accordingly. Going in on the super resolution microscope, the first thing that I wanted to look at was if the dyes are attaching in the way that we were expecting them to. And so if you look at this image, it is very, very dim, and I was actually looking at the long-Stokes shift dye, specifically in the red channel. And so what we were expecting was for this intensity to be a lot higher, which would indicate that the structures are showing up in red, which is what the long-Stokes shift dye's purpose was to be. In actuality, it was not attaching in the way that we were expecting it to, and you can see that the intensity count, or the brightness count, was 18. When I used the green detector in the green channel, I was able to determine that we are seeing it in the green with a lot of brightness and intensity count of 900. And it's also able to be steaded, or the super resolution microscope can resolve those structures very well, showing that the dye was not attaching in the way that we were originally expecting it to. And so when I went in, I also looked at those pre- and postsynaptic proteins that I was talking about, which would be binded to the axons and the dendrites, and you're actually able to resolve them very well on the STED, or the super resolution microscope. And so these were stained with just typical dyes. These aren't long-Stokes shift dyes, which shows us that this microscope, the STED, works very well for resolving these structures, but our dyes were not attaching in the way that we wanted to. So I started looking at where there was the potential for this staining procedure to go wrong. And so I actually went back and took a look at that wide field image that I took on the confocal microscope. And if you look, you can actually see clusters of dyes that aren't necessarily binded or conglomerated with the different structures that were in our regions of interest. When I went to the super resolution microscope, I was able to resolve those structures and see that these were actually our clusters of dye, and that they weren't attaching to the structures in the way we were expecting them to. Going forward, we determined that the best way to figure out where exactly the problem was to look at a different tissue and stain it with these long-Stokes shift dyes. If we were able to resolve the structures that were stained with these long-Stokes shift dyes, it means that there was just an issue with the staining procedure for this sample, and we can proceed with a new staining procedure in order to differentiate between the structures and see if we can look at those points of communication. If we're not able to go in and see these structures with the new dyes, that means that there's an issue with the dye itself, and that we can go ahead and communicate with the company in order to determine the next steps and potentially look at other long-Stokes shift dyes. So I would like to go ahead and thank Dr. Nikolai Urban and Dr. Mary Phillips for being there every step of the way and teaching me how to use these different microscopes and breaking down really complicated concepts so that I could apply them to my experiments. Dr. Ilaria Drago for organizing this entire program and being there every step of the way and checking in with us and making sure we were getting acclimated and that we were staying on top of our projects. Dr. Juliana Yapel for providing these samples and also providing information for me regarding the actual experiments that were going on so that I had the correct neuroscience applications. Katie, Shirley, for being there every single day and guiding me through the experiments and guiding me through everything and helping me to answer some of the questions that I was coming up with. The other interns for making this such a memorable and fun 6 weeks, and the MPFI supporters for making this program possible.
Great. Any questions for Alana? And then there is one in the back.
It's been 1,000,000 years since I've used a microscope. How do you take your sample and at that level and go from machine to machine and still be in the same place? How does that work?
So, well, actually, the question was, how do we go from machine to machine and go through these different samples and know that we're looking in the same region? So I know that I'm looking within the visual cortex because we're looking at different visual stimuli in order to determine the effects of these stimuli on neural connections or the synapses. And so sometimes we'll actually go in and mark using a very high-powered laser so that there might be little burn marks or something so we can find specific regions and go back to them when we're going between microscopes. And then I'm also just looking at connections within the visual cortex, and after determining that region of interest, if it's not a specific synapse that I'm looking at, I can actually just go in and look at any axon or any dendrite just to see the generalized connections between them.
Any other question? In the back.
Hi, so you said that you used a super resolution microscope called the STED. How exactly does that work in resolving such details on the structures?
So the question was, I used a super resolution microscope called the STED, and how does that go ahead and resolve these structures? So first I'll refer to the confocal microscope that I used, and we would use in the confocal an excitation beam to excite those fluorophores or fluorescent proteins that I referred to in order to visualize these distinct structures. But the thing with that is that we're not able to see structures that are smaller than 200 nanometers because we're getting a lot of noise, and we're getting a lot of out of focus light, and we're also activating some proteins that we don't want to be activating. And so the STED, or the stimulated emission depletion microscope, has a second laser, a depletion laser, and that laser goes in and tells certain fluorophores that aren't necessarily what we want to see, that they're noise, to turn off, so they'll release their energy without actually releasing a photon or a particle of light, so that we can differentiate between structures and see things that are smaller than 200 nanometers.
Any other question? Okay, thank you so much.
Thank you.